Good morning, everyone. Good morning, everyone. Today on National Science Day, we at TIFR Hyderabad are hosting virtual tours of three of our labs on campus. All these tours were conducted under strict COVID-19 protocol. Each lab tour will be followed by a live Q&A session where the audience can interact with the researchers and know more about a day in the life of a researcher. You are requested to post your questions on the live chat. So, curious to know more about how we go about addressing a scientific problem? Well, you don't have to wait any longer for this answer. Here is the first lab tour of the day, the Quantum Materials and Quantum Transport Lab. This lab, led by Karthik V. Raman, makes new materials and studies their quantum properties.
Hello everyone, my name is Karthik and I'm a scientist here working in the field of experimental physics. So before we take a tour of this lab, let me give you a brief perspective about the kind of research activities that we do here. Now, all of you are familiar with a periodic table. Basically has elements which can be of various kinds. It could be solid, liquid, gas, or it could be categorized as metals, semiconductors, or insulators. Now what we do is to bring all these elements together. We pick and choose these elements to form compounds, and these compounds are special compounds which we can also call as quantum materials. And the application of these quantum materials is primarily to explore new fundamental opportunities in terms of its physics and also translate it into technology which is applicable in anywhere in the industry today, whether it is in terms of your gadgets as your smart laptops, your smartphones, or your computers, or it could be something very sophisticated for defense or military. Now, all this work that we do here is based on a concept of thin film technology, where we don't really make bulk materials, but we try to engineer with atomic precision the arrangement of these atoms on surfaces and thereby try to probe its properties, which could be electrical, optical, or magnetic. And all these three properties has its own merits and applications, as I just mentioned. So with this, let us start the lab tour, and my students will explain you the various lab activities that is going on here.
Hi guys, this is Quantum Materials and Transport Lab of TIFR Hyderabad. This is headed by Karthik Raman, and a lot of grad students and postdocs carry out their research here. And today we will be showing you our lab and what happens in our lab every day. So come on in and let's have a look. So this is Archit, who is a grad student here, works on growing quantum materials, and today we will learn from him what he does using his interesting equipment.
Sure.
So what are you doing and how does it work? Can you explain it to us?
Yeah, so this is the MB and it stands for molecular beam epitaxy, and this helps us to make thin films out of any material that you can choose from the periodic table. So these thin films that I'm talking about, they are very thin, and if I can give you an idea how thin it is, and it's about 10,000 times thinner than a sheet of paper. And the reason why we are doing this thin film study is because if you look around, if you look at the gadgets that we have in our everyday life, so for example, mobile phones, smartphones, and laptops, they are all made out of microprocessors. Now these microprocessors are the result of the experiments and the advances in the physics for last few decades. But still we do not understand it to the full extent, especially with the materials that have come up in the very recent times. And we need to really do experiments to know more about them. But before we start doing experiments, we have to make these films, and this has a control of about this. You can control it down to a single layer of any material that you can choose. For example, a metal insulator, or if you talk about more exotic materials such as superconductors, and the more recent materials such as topological insulators. So you can make these films here, and then you can start doing experiments with it.
So you say that you can know materials atom by atom.
Yes, so you can make stack another atom layer on top of each other. So that's the beauty of this instrument. So you can have that much of control. Yeah.
Can you show us your equipment?
Yeah. So how do you make thin films? It's basically you need a substrate. You need something on which you want to deposit, and then you need a holder on which you can mount the substrate. This is the holder, and this is the substrate mounted on it. And then what you do is you put it inside the chamber, but it has to go through it has to go through a number of chambers in between. So this is called a load lock, and you mount it here. And this chamber is a small one, so it's relatively easy to pump it, and this can be done without breaking the vacuum of other chambers. This is how the journey of a sample starts. And then you can, it has some arms, mechanical arms, which you can have a control on, and it can move to the MB. Now, inside the MB, what you do, so you can deposit these materials by evaporating them, and the way you evaporate is basically just heat it up and then let it boil, and just go up in the air, just like water boils. Yeah. So there is a catch here that you cannot do this experiment in open air. That's because when the atoms are going and sticking on the substrate, there are a lot of dirt and other unwanted materials that can also go in and stick to it, and that basically can make your whole film bad. So the way you do it is basically perform the same experiment in the vacuum, and to create vacuum we use a series of turbo pumps. As you can see here, there is one turbo pump here, and it basically sucks out all the air inside the chamber, in this chamber right now. So I'll show you the various parts of the MB. So in this part is where all the sources are kept, and this is where it heats up and goes and deposits on the substrate. And from here there is a viewport here, glass viewport, through which you can see the sample and maneuver it in whichever way you want. So there are water lines here which keeps the body cool, because you're heating a source, so you need to keep the things around the source cool, otherwise they will also start evaporating. And there are a lot of materials inside which has different evaporation temperatures, and there are two ways we can evaporate these materials. One is to just heat it by coils and then let it evaporate. But then there are few materials which has much higher evaporation temperatures, for example metals. So what we do there is we bombard high energy electrons and heat it up to extreme temperature so that it eventually starts evaporating. And once all these films are made, you can bring it out and start experimenting with it. So if there is a need to evaporate a material which is even higher temperatures, there is a method called sputtering that will be discussed later in this video.
without breaking any vacuum here. And we have Suman there who will show us how a sputtering unit works. Suman, can you show us?
Okay, this is just the basic schematic of the sputtering. This is the gas inlet where we float the basically inert gases into the chamber, and this is the substrate holder where we will mount our substrates, and this is the target of the element which we want to deposit on our substrate. Okay. Then a high potential is provided in between your target and your substrate. Then the inert gas present here will dissociate into positive and negative ions, and these highly energetic positive ions will knocks out the atoms from the target surface and gets deposited on the substrate. This is just the simple mechanism of the sputtering, and I have shown just all these things by this schematic. Now I will show you how these things are done in our actual chamber. Let's go there and see. This cubical chamber is our actual sputtering chamber, and these are the gas lines from where the gas is flowed into the chamber and flow is maintained. This is the turbo pumps for the vacuum creation, and I have already mounted the substrate inside the chamber, and I have already maintained the argon flow. Now we have to just switch on the power supply and create the plasma. Okay, let's go there. Now I am just switching on the high voltage. You can see this current is coming. This is basically plasma current. This is around 123 milliamp. Now we can go in the chamber and check our plasma. There is the sample we have mounted in the load lock, and this purplish color is plasma.
So thank you, Suman, for your explanation of the sputtering. So if we have to take a one-liner, sputtering is a technique where materials are evaporated using plasma technique. Now let us move on to the scanning tunneling microscope, which is on the other part of the lab. Come along. Here we go. So this is a scanning tunneling microscope here, which will be explained by Karthik, and it's up to Karthik now.
Okay, so now what you see here is an instrument called the scanning tunneling microscope. So the purpose of this instrument is primarily to image surfaces with atomic resolution. And how we do this is that we start loading a sample from the load lock, which was explained earlier, and we grow our samples, films, in the chambers, and finally we move it to this microscope. And what you do here is you have a tip, it's a tungsten tip, which is very close to your surface, and it's at the level of few angstroms. And we make this tip scan over the surface to primarily look at the atoms and look at the properties of these atoms as a function of applied magnetic field or applied bias. And this essentially gives us very exotic inferences about the classes of materials that we are probing, whether they are semiconducting, magnetic, or non-magnetic. So, so with this, I can explain on this computer here how the images are taken. Okay, so this is an image of a surface of graphite, where what you see here are nothing but arrangement of atoms, periodically arranged essentially. And each spot, the yellow bright spot, are the carbon atoms. And you can see they form a hexagonal structure, which essentially gives you an impression about the kind of arrangements through which these carbon atoms are placed in your graphite sheet. So this instrument, I explained to you that it essentially allows you to image surfaces with atomic resolution. Now, one more capability of the instrument is to be able to go down in temperature. You can go down to extremely low temperatures, which helps in improving the image quality, because now the atoms no longer can vibrate. When you go down, when you cool down your system, your material, the atoms in the material cannot vibrate much. And that essentially improves the quality of your imaging. That's one of the things that is special to this system, where to be able to go down to this low temperature, you don't need any liquid cryogenics. You need typically liquid nitrogen and liquid helium, which are very expensive to be able to really cool down your system and maintain at low temperatures for a very long time. But with the facility that we have right now, we really don't have to worry about these liquid cryogenics. We can go to space temperature using a cold head technology and constantly measure 25/7. So now the instrument here that we have is prone to any source of external noises. And noises can be of various kinds. Mostly it's acoustic, it's electrical, and it's vibrational. Now we have made sure that we can decouple all these three sources of noise by a judicious choice of the design of the system. Now let's start with the vibrational noise. So we have this instrument which is housed on a big chunk of concrete mass. It's almost 50 tons of concrete. It's sitting on sand and completely decoupled from the building foundations. And this ensures that even when someone walks outside on the service corridor or really walks with something, a very massive piece of equipment, you really don't see any noise on your images that you acquire. You can still see your surfaces with atomic resolution. The other source of noise is the electrical noise, which I explained. The electrical noise comes because when you have any instruments running across the other parts of the chamber, it can give rise to line frequency noise, which is of the order of 50 hertz and its harmonics. You can decouple them by using a technique which is it's called an electrical isolator. It's nothing but a piece of ceramic insulator that decouples the two grounds of your system. And it's therefore using this technique that we have adopted with this complete integration that makes this overall setup unique in the country and also in the world in terms of the capability of imaging, growth, which can be done simultaneously without any interruptions.
So thank you, Karthik. So if we learn about this, scanning tunneling microscope basically images surfaces of metals, and it gives you pictures of atomic resolution. So now that we have grown materials and looked at them, we can also further go and measure their electronic properties. And there is a tool to do that, and Satyaki will explain us exactly that. So let's go to the next part. So here is Satyaki. Hi.
Hi.
And Satyaki will explain to us what is this interesting thing he is doing.
After preparing the samples, we use this instrument to measure different electronic properties of those samples, like electron density, hole density, resistivity, mobility, conductivity, this kind of things. So before measuring any of these properties, first we need to remove all the heat from the sample to remove any thermal noise coming from the vibration of atoms. So this chamber is basically a refrigerator which cools down the sample up to very low temperature, almost close to absolute zero. So this is a compressor that repeatedly compresses and expands helium gas, and doing this thing repeatedly, it cools down the sample. So if you see this thick pipeline, so helium gas comes to this line and goes away. So if you see this stick, so this is our sample, and we put this sample at the end of this stick, and this stick goes all the way to the bottom of this cylinder. Okay, and if you see these cables, so these cables are connected to the sample and to the controllers here. So these controllers basically measure temperature, current, and voltages. We also measure these electronic properties of our samples at different magnetic fields. So we have a big magnet inside this chamber. These controllers are connected to a computer here, and all of our experiments are automated. You can see these data plots. These are basically a measurement that is going right now.
So far you have seen a molecular beam epitaxy unit which grows ultra-thin films, a sputtering unit, a scanning tunneling microscope, and a dilution refrigerator which goes down to really low temperatures. So this was a virtual tour of one of the most exotic state-of-the-art labs in our country. If you are interested to come and visit our lab and have a look at the facilities here in person, you're most welcome. Please get in touch with us via email and we'll be happy to respond to you. And thank you for your attention, and I hope you all enjoyed having a look into our lab. The floor is now open for questions, and most of our lab members are here, and you're most welcome to ask any number of questions that you like.
So now that we have toured the first lab of the day, let us take some questions. So we have one question. Why do you want to use helium gas and not any other gas in the cryostat refrigerator? Anyone from Karthik's lab can choose to answer this.
Okay, let me answer. This is Karthik here. So that's a good question. It depends on the temperature that you want to attain. So if you want to go to say around just a temperature of around 100 Kelvin or 77 Kelvin, you can use nitrogen and you don't need helium. But liquid helium so the helium turns liquid at a temperature of around 4.2 Kelvin. That's around minus 270 degrees Celsius. So if you want to cool down your sample to that temperature, you need some kind of cryogenic liquid that stays there and cools down your system. So at the end, the choice of gas or choice of material is based on the questions, based on the temperature that you want to attain on your sample. So liquid helium is good if you want to really reach temperatures of the order of 5 Kelvin to 10 Kelvin. And there are no other gases typically that are liquid in a liquid state at such low temperatures and easily available for measurement. At the same time, you want these liquids to be electrically not conducting, like insulating. That's the other thing that you need to worry about. So overall, the choice is any liquid, any gas that can stay in a liquid form and be stable at low temperatures is what we need. And helium is having all those essential criteria to go down in temperature and do those measurements. Yeah, thank you.
Okay. Thanks, Karthik. I'll just repeat the question maybe.
Yeah, are there any other questions?
Yeah. So how do you know that you have deposited one atomic layer of the material?
Okay. So the measurement of thickness is primarily done by using a microbalance inside the vacuum. So it is nothing but a quartz material which has a resonant frequency of its own acoustic vibrations. And what we do is when a material is grown on this quartz substrate or the sensor, its resonant frequency changes because of the addition of extra mass on the surface. And the change in frequency directly correlates with the material being grown, and one could essentially attain precision of the order of subatomic level. So you can really look at the growths which can happen at the rate of 1 angstrom or even 0.1 angstrom per second. So that is how we use. There's a microbalance using quartz which tells you the rate at which you're growing your films. So I hope I answered the question. There's some other questions which I see. The second question is, in a sputterer, what are the kind of gases you can use to generate plasma? So that's a good question. So one could use, typically there is no limit on the gases that you want to use. Argon is used primarily an inert gas to create the plasma and to hit the target, to bombard the target and essentially create atoms that can be grown. But sometimes you want to even start with the elemental target and make compounds. And that can be done by even sending, reactive gases along with argon. This could be oxygen or nitrogen or any other reactive gas to form, for example, if you put in oxygen along with argon, say in a material like titanium, you can create titanium oxide on your substrate. Same way you can create nitrides. There are compounds which are nitrides, which one could grow them as films, starting with elements as targets. And this is a technique called reactive sputtering, where one gas is reactive in nature, which interacts with the atom that is created, and finally forms a compound on the sample. So I think that's essentially tells you that reactive gases can also be used depending on what compound you want to make. So I hope I answered this the question. And the third one is, yeah, so that's I've already answered about the helium gas being used as the cryostat refrigerator. Thank you.
Yeah, thanks, Karthik, for clarifying the doubts. So the second lab tour of the day is of the collective cellular dynamics lab. This lab, led by Tamal Das, looks at how forces help the biological cells to work and survive together.
Hi, welcome to Collective Cell Dynamics Lab. In this lab, we are interested in understanding how forces affect the biological processes. Especially, we are trying to measure the forces which are exerted between the cell and the extracellular matrix and between two cells, and we are trying to understand that how these forces in principle affect some of the biological processes like collective migration of cells that happens during wound healing, or the cell competition, which is at the very basic level of how our tissue maintains its quality and gets rid of the cancerous cells. In addition, we are interested in understanding that in a sort of broader way, how the tissue mechanics influences different biological processes. So for this, we use an extensively cell culture, microscopy, as well as a lot of biophysical tools. Why don't you take a tour of the lab now?
So let us take a tour of the CCD lab. Okay, that's very nice to welcome us. CCD sounds like Cafe Coffee Day, but I think it stands for Collective Cellular Dynamics Lab. And it says, please remove shoes before entering. So now we are entering the cell culture. Hi.
Hey,
Are you busy?
yeah, just give me 2 minutes. You can come in, but give me 2 minutes.
Okay.
Yeah, so tell me.
Hi. So I was looking around while you were busy adding things to certain plasticware, but I couldn't make out what actually do you do here. I know it's called cell culture, but what is exactly that you're doing here?
Okay, so as you read outside, this is the cell culture facility. And what we basically do in a cell culture facility is basically culture cells. So what I mean by that is that as humans are growing, similar to that, we need to grow cells, right? And to do that, we need certain specialized equipment, certain specialized material, which we basically keep here, and then we culture cells.
So you switched on something on this chamber. What was that?
So this is a laminar hood. So, and what I just switched on was ultraviolet light. And why I did that was to basically maintain a sterilized environment. So UV sort of acts as a killing agent So must, just like how we are taught in our school days that if we are hit by the sun's UV light, then we'll probably die. So similar to that, what we do is we maintain a sterilized environment, and for that we switch on the UV.
And I also saw some solutions, ethanol or alcohol.
Right.
Yeah.
So this is one way of doing that. There, of course, are other means of maintaining a sterilized environment. So another way we do is basically use 70% ethanol solution. It's similar to what people are nowadays using sanitizer for. So it basically acts as a disinfectant, and it kills any microorganism which might be populating our hands and other areas. So to clean out our benches, to clean out our hands, to clean out the surfaces of whatever we are going to use, we disinfect with 70% ethanol, and that is what you saw me spraying around.
And what are these big, something that I think looks like a refrigerator?
Oh yeah, no, so these are not really refrigerators. Refrigerator is the one you see there. These are what are called incubators. What they basically do is they provide cells with such an environment so that they can grow and flourish. So for us, this environment is enough for basically our growth. But to maintain cells, we cannot let them just grow like that. So they require certain kinds of growth conditions. One of the most important growth condition is the temperature. So what you see here, this temperature in degrees Celsius, this is maintained at 37. So this 37 degrees Celsius temperature is optimum for cell growth, and that is what these machines here do. On top of that, what cells additionally require is some CO2 concentration. The one which is present in the air is not sufficient for them to grow. So what this machine does is that it is attached to these cylinders here, you see, right? These are carbon dioxide cylinders, and what they do is through these blue pipes they pump carbon dioxide to these incubators, and they have a certain system through which they maintain carbon dioxide inside at a level of about 5% air pressure. So these two things are the most important conditions which cells require to grow, and that is what we maintain.
So I saw you add a red colored liquid to those plastic ware. What was that?
Oh, so that liquid which you saw me adding was basically what is called a culture medium. So what it is basically food for cells to grow, and that is what I was adding to the plasticware which you saw me earlier. So before I go take out the medium, I'll sprinkle some of the disinfectant so that we have sterilized our hands. This is what is called a culture medium, and this is the red colored fluid which you saw me adding earlier. What it has is basically components which are required, chemical compounds which are required for cells to grow. So one of the major things it has is glucose, because it is the carbon source which cells utilize to grow. Other than that, there are vitamins, there are other amino acids which are essential for cells to grow. The red color is imparted by a dye which is added just to showcase the health of the cells, which is called phenol red. So what I did earlier was splitting cells. So what I mean by splitting cells is basically, I can maybe show you with an example. So here I have a culture flask, you see, and this flask is basically where cells are growing and flourishing. You see the bottom. If I just compare it against the light, you might be able to see that the bottom region is a bit cloudy. So that is what this is how cells would look like under light. And how they grow here is basically what they spread throughout this surface which we have provided them. So once they have covered the whole surface, of course, if we need the cells to grow further, we of course need to give them more area, right? So either we can increase the surface area on which they are growing, or what we do is called splitting cells. So what we do is we basically take them off the substrate, we collect them, and then we add it to a fresh flask where they can flourish next. And by this way, what we can do is basically maintain cells in culture and then use them for our experiments.
Okay.
So
can I have a look at those cells? Is it possible?
Of course.
With the naked eye?
So with the naked eye, you'll be able to see this cloudy thing. But if you really want to see cells, what we have here is a microscope, and I think you'll find it more fascinating if you look at it under the microscope. So let's go on the microscope and have a look at it. So yeah, so here you go. So now here what you see is basically cells that are 4 times higher magnification. So you're seeing them as 4 times the size they actually are. And this is the lowest magnification this microscope will offer right
So
now.
can we go at a higher magnification?
Yeah, we definitely can. Next we can maybe try a 20 times magnification and let's see how it goes.
So this is at 20x.
This is at 20 times the magnification.
And
yes.
I can see some bright lines, right? Are these the boundaries?
Yes, so these are the boundaries shared by neighboring cells.
Okay.
And that's how these particular cell lines, these particular cells grow.
And the highest you said was 40x on this.
Yes, the highest would be 40x. So let's go to 40x. So now what you see here is a magnification of 40 times the actual size of the cells.
This is actually quite fascinating. I see some circular structures now.
Yes, so what you see, the bigger circular structures inside each of these cells is the nucleus of the cell. That is sort of what acts equivalent to what our brain does to our body. It basically controls the major functionality of the cell.
But there would be other structures, and will it be possible for us to magnify them further and have a look?
So there are definitely other structures than the nucleus in these cells, but as of now we won't be able to magnify it further with this particular microscope. But if you are still fascinated and you want to look at the other structures, you can actually talk to one of our microscopy experts, Satya.
So we are now going into the microscopy room. Satya, are you busy? Can I talk?
No, it's okay.
Yeah, so I was talking to Pravar, and he showed me some cells under a microscope. They look really good, but I didn't follow, how the microscope works.
Okay.
So can you help me out?
Yeah, of course. An optical microscope is an instrument which developed in such a way that it can produce magnified and resolved image which we can't see with our naked eyes.
So you said magnified and resolved. Are these different?
Yeah, different. As you heard about that, in December 2020 there is a astronomical event called as the Great Conjunction of Jupiter and Saturn, where Jupiter and Saturn came close to each other. So on that day I click a mobile picture. The left one is the mobile picture and the right one is taken by a telescope. On that mobile picture, I just zoomed the Jupiter and Saturn, and I have observed that there is only one spot I have observed. But in the case of telescope, we have observed two points, Jupiter and Saturn, both with distinguishable manner. So this happened because the telescope have a higher magnification also and a higher resolution. In microscope also there is a device called as the objective lens. Here you can see there is several objective lens with several magnification, and this objective lens help us to magnify the specimen as well as to provide a higher resolution image of the specimen. There are several other important parts of the microscope also. Here is the microscope, and this is the halogen lamp of the microscope, and it provides the illumination light to the sample plane. And here is the sample stage where we can put the specimen under here, and under the sample plane there is a objective turret. In that turret several objective we can mount there, so that we can image the sample with different magnification and resolution. Okay, then the light goes down, and then there is some beam splitter so that it splits the beam towards the eyepiece. Here we can observe the biological specimen here, whether it focused or not. Here we can focus by this focus now, and also another part of light gets deviated towards another port of microscope and goes towards the camera, and in the camera we can capture the image of the sample placed on the microscope stage.
The microscope I saw in cell culture was a little different than this one.
Yeah.
So are there any differences in microscopes?
Yeah, there are several types of microscope based on illumination path or detection path. So based on illumination path, The microscope can be divided into several types: wide field microscope, confocal microscope. Okay. Based on the detection path, the microscope can be divided into several types, such as bright field microscope, reflection mode microscope, and fluorescent microscope. In bright field microscope, the transmitted light from the sample get detected by the detector, whereas in reflected light microscopy, the reflected light from the sample get detected by the detector. Before going to the fluorescent mode, let me explain the fluorescent phenomena. Some molecules in nature, when it illuminated by a particular wavelength or particular color of light, let's say blue, it get the blue light is absorbed by the molecule and it emit another wavelength of light, let's say green. The green light is emitted from the molecule, and that we use that green light to detect the molecule. We use that green light in the microscope. That is called the fluorescent microscopy. Now let me show you the principle of working principle of fluorescent microscopy. Here a source of white light gets incident to the excitation filter, and from excitation filter a blue light is reflected by the dichloric mirror and gets to the specimen, and from specimen in a fluorescent light, let's call the green light, is is coming from the specimen and transmitted by the dichloric mirror and emission filter to the detector for detection purpose. Okay, and these excitation filter and dichloric mirror and emission filter, and these three in microscope is combinedly put in a fluorescent filter cube. Yeah.
Do you have a filter cube? Can you show us?
Yeah, we have a filter cube. We can you can see this is a filter cube, and this means this is the dichloric mirror, and this is the emission filter, or this is the excitation filter here. We take a white light and pass through the excitation filter, and it reflects by the dichloric mirror and goes to the sample plane. And the sample plane, you can see in the paper, the blue light is going to the sample. And if the sample emits green light, then it passes through the dichloric mirror and emission filter to the passes through the detector to detect.
A very cool technique, Satya, but I was wondering how would you use this in biological samples?
Yeah, in biological sample, as we explained that the molecules, the fluorescent molecule, we can tag them different structures in the biological sample so that we can image by the said technique, the fluorescent microscope technique.
So I saw some structures in 40x objective, but
yeah.
Now if you want to look at detailed structures, how do we do that?
Yeah, we have a set of objective lens like 40x, 60x, 100x, and with different magnification, so that we can image more detail into the cell. But there are certain limitations of the microscope, so that it cannot go beyond that limitation. That is called the resolution limit. And for going beyond the resolution limit, there are several techniques, such as electron microscopy techniques, and for optical microscope there is super resolution microscope technique, but they are tedious and very costly for implementing that one. But in our lab we have a very cost effective and convenient way to explore the minute detail that goes beyond the resolution limit. That technique is called that expansion microscopy. You can ask Simran or Prabha for that one.
Hi Prabha.
Oh, hi again.
So Satya told me about resolution and magnification, and he mentioned a very interesting technique called expansion microscopy.
Right.
So could you tell me about it?
Yeah, definitely. So expansion microscopy is one of the recent techniques which achieves super resolution, which is not achievable by conventional microscopes. So what expansion microscopy basically does with the cells or the specimen is basically it physically expands the cell so that we can achieve greater resolution. And how we do that is basically we embed the specimen or the tissue or the cells in a resin. And funny story, that resin is actually inspired from something which is used in diapers, and it has a property wherein it can absorb water and it expands. So inspired from that, what people have developed is that they use the same resin to embed the specimen and then physically expand using water. So what we essentially do here is that we expand the specimen, the cells, and then we are able to resolve the structures which are not resolvable by conventional microscopy. Since you are interested, you seem to be interested in that, my colleague Simran is right here, and she's already working on one of the experiments with expansion microscopy. And why don't you go ahead and talk to us?
So now we are going to Simran, who will tell us about expansion. Hi Simran.
Hi.
So Pravar told us that you do a very cool technique in the lab called expansion microscopy.
Yeah, so I am doing expansion microscopy today, and so I think Pravar has told you the principle and everything, that why are we using expansion microscopy to get better resolution and see the internal structure of the cells. So basically the protocol involves 4 major steps: fixation, and then embedding of the sample or our cells into the gel, and which is going to expand, and next step is the homogenization, and last and the final step is the expansion. And I'm going to show you each of the steps right now. So this is the petri dish. You have seen how we culture the cells. I have taken it out of the incubator. I'm going to aspirate the media out of it and add the fixator and fix the cells.
So what is the purpose of this fixing?
So basically what we want is we want to preserve all the internal structure of the cell. So what this fixative is going to do is arrest everything that's happening inside the cell and preserve each and every internal structure as it is. So now we are going to keep the cells with the fixative for 15 minutes, and then we'll wash and move to the next step, which is the embedding. Okay, so 15 minutes are over. We have washed the cells, and we have taken out the cover slips and kept it here. Now we are going to move to the next step, which is embedding. In this step, basically we have to embed the cells or our sample into the gel or into the monomer solution and let it polymerize and solidify around our sample. So how we are going to do it basically, so this is the monomer solution. I'm going to add 2 drops of the monomer solution over here, and then I'm going to put these cover slip which contains my cell onto the solution. Now we are going to keep this at 37 degrees for 1 hour and let it polymerize and solidify, and then the cells will basically be embedded into the gel matrix. So it's been 1 hour, our gel has polymerized and it has our sample in it. Now we are going to homogenize the sample before we add water and expand it.
So your cells are already embedded in the polymer mix, right? So what will happen if we just add the water now? They should expand, right?
Yeah, so in principle, if I add water to this to these gels, they should expand. But the problem here is that we have cells in here, and in case of cells, there are some places which are probably more dense as compared to the other places inside the cell, and in that case the expansion will not be even. Some parts will expand faster and more, and some parts will expand slower and probably less, and we don't want that. We want an even expansion of the whole sample. For that, we are going to do this step where we are going to put these gels in a homogenization mixture and put it at 70 degrees for 1 hour, and then we are going to do the final and the most important step of the protocol, which is adding water and expansion. So I am going to gently remove my gels, which are, you can see that they have solidified, and put it here. Now this 6-well plate contains the homogenization mixture. I'm going to add and put my gels in this, and then keep it at 70 degrees for 1 hour. Okay, it's been 1 hour now, and I have taken the gels out of the oven, and I put them on these petri dishes, and you can already see that they have expanded a little bit. Now we are going to do the final step of expansion, where we are going to add water. Yeah, so I'm going to add excess of water. And now we will see in 30 minutes that the gel will be expanded. So basically we are going to do 3 rounds of expansion, 30 minutes, and then 1 overnight expansion, and by the end of it we will see that the gel will expand to 5 times of its original size.
So I just finished my tour of the CCD lab, had a really fun and exciting time, learned a lot of new things, and I think you guys also have a lot of questions, so please feel free to ask them.
So we have some questions in our live chat box. The first one is from Athira Murli. The question is, as a result of cell respiration, carbon dioxide is expelled. Then why is it again supplied into the incubator for cell growth? So anyone from Tamal's lab can answer.
Yeah, so I'll take this question probably. So that is an excellent question, Athira. And I agree with you. Cells do respirate and they release a lot of carbon dioxide while respirating. But considering the volume of the incubator, that carbon dioxide is probably not enough to maintain that 5% air pressure, which I just mentioned. So to maintain that, we need to externally pump some carbon dioxide in the incubator as well. So that is why we do that. I hope that answers your question.
Yeah. The next one is from Ryan Pathak. What was the red culture medium and what type of cells does it support?
Hi, Ryan. That's a good question. The red culture medium, which I showed a while back, was the growth medium, the growth food for the cells. It contains all the nutrients, all the vitamins, all the minerals which the cells require to grow and flourish. And what kind of cells it supports is, in our case, the medium which I showed, it can support mammalian cells, so cells which are derived from mammals. If you need to culture some other cells from some other type of organisms, you will probably need some specialized other kind of culture medium. That one is optimized for cells derived from mammals. Those kind of cells can be grown with that medium. So I hope that answers your question.
Yeah, thanks. Now we have two questions from Cryptic Drosophila. First is, what's the difference between wide field and confocal microscope? And why is the dichroic mirror reflecting blue light and transmitting green light?
Hi, probably I will take that questions. So the first question is the difference between wide field and confocal microscope. In wide field microscope, the illuminated light means illuminates the sample volume at totally entire area of the sample is illuminated by the illumination light, and in the same way the entire area of the entire sample volume is detected by the detector, so that there is some out of focus light coming from out of focus region detected by the camera or detector. But in the case of confocal microscope, each point of the sample, each point of sample is illuminated and imaged separately. At the final, in the final, we combine all the image points to get a higher resolution image in the confocal microscope. Hope this will get clear. And I'm coming to the next question. We in our this YouTube, sorry, this lab demonstration, we first went to Karthik's lab, Dr. Karthik's lab. In Karthik's lab, they used thin film technology to grow thin films on some medium. In dichloric mirror, what happened? In dichloric mirror, they deposit several layers of thin films on a glass plate, so that when light incident on that glass plate, interference happens, and selectively some wavelength get transmitted and some wavelength get reflected. In our case, the blue light and the thin film are deposited in such a way that the when an incident take white light, the blue light gets reflected, and means other part of light get transmitted. Yeah, hope this will I can answer the question.
So we have a question from Avantika Rai. Can expansion microscopy be used for both adherent and non-adherent cells?
Um, so expansion microscopy is not limited to any particular cell type or anything. In principle, you could apply it to any kind of cells and even tissues. And people have done experiments where they have expanded brain tissues and other tissue and different type of cell types and expanded the samples and seen the proteins and other biomolecules with a better result.
The last tour of the day is that of the Materials and Interfaces Lab. This group, led by T. N. Narayanan, is finding ways for tuning interactions between atomic layers for energy applications.
The Materials and Interface Engineering Lab works on three different aspects: energy storage, growth and application of 2D materials, and photoelectrocatalysis. Now, the lab members will walk you through.
Hi all, myself Narayanan. I am a faculty here at TIFR Center for Interdisciplinary Sciences, Hyderabad. And our lab for the last few years, say example 5 to 6 years, working on various aspects of energy. This includes energy storage, harvesting, and so today I'll be explaining how do we do some of these energy storage devices. So you have all seen batteries in various forms. So our students Pallavi and Amar will be demonstrating how a very common lithium ion battery that you can see in all your portable electronic equipments work, and how do you develop these kinds of lithium ion batteries in lab. So for example, this is a lithium ion chip cell, and this has a voltage of 3 volt, and this is works really as a battery, as you see in your portable electronics. And now Pallavi and Amar will be demonstrating how we can really make these kind of batteries from scratch.
This is the working mechanism of lithium ion battery. What you see in orange is a negative charge anode, in green positive charge cathode, in blue is a electrolyte, in gray is a separator which prevent the contact between both electrodes. During discharging, which means we are drawing power from battery, in that anode lithium get oxidized and generate electrons which flow from the outer circuit, whereas lithium ions move from anode and intercalate in cathode. During this process, current is generating and you can see the light is glowing. So as Tinan told, lithium ion battery is a highly interesting research topic because of its high specific capacity and the discharge voltage. As we know that the first commercial lithium ion battery came in 1991 by Sony. After that, researchers are trying to improve their capacity by changing their by improving their components and the electrolytes as well. So here we are demonstrating the lithium ion battery as a half cell. So battery assembly is done inside this argon-filled glove box where oxygen and water content is very low. Our components are already inside this glove box, and they are positive case, a graphite cathode, separator, lithium anode and electrolyte, a spacer, spring, negative case. Let's start the assembly. Here we are keeping graphite electrode on positive case first. Then we put separator to avoid contact between two electrode. Then we are putting lithium rich electrolyte. After that we are putting lithium metal, then a spacer and ring to make better contact between two cases. Now I am putting negative case. Now I am closing it by finger to ensure that battery is sealed. We are applying hydraulic pressure. Let's take it out our sample. Battery looks fine. Let's check their OCV, and its OCV showing 3 volt, which is good. And to check further, we are putting on a holder, and you can see this LED is glowing. It means it's working.
Hi guys, you have already seen the assembling of lithium-ion batteries which you use in your mobile phones, toys and gadgets. Lithium is one of the most reactive elements that we know today, and in addition to this, the organic electrolytes that we use for the assembling of lithium-ion batteries, they are prone to combustion. So this makes the whole system very unsafe, and it's difficult to travel with these batteries. So there are what could be the other alternatives which could replace these lithium-ion batteries. These include the aqueous electrolyte based batteries which has electrodes such as zinc, aluminium, iron or copper. So I want to point out one more disadvantage of lithium-ion battery, which is its low energy density. Now you can think energy density in terms of energy stored in unit weight of the sample. If we think about all this, there comes another category of batteries that could actually replace the lithium-ion batteries. This category is the metal air battery, where one of the component of the battery is replaced by the air itself. So we get a high energy density which could run our vehicles or cars without consuming lot of material or inbuilt reactants. So today I'm going to demonstrate you the assembling of metal air batteries. Now I'm going to show you the main components required to assemble a metal air battery. A zinc anode, a separator, electrolyte and a platinum coated carbon paper, which is the cathode. Now the zinc anode, we'll put it first, which act as a negative electrode, and then we'll put separators to avoid the contact between two electrodes. Now we'll add few drops of this alkaline electrolyte, which is 6 molar potassium hydroxide. We'll put the second electrode, which is the cathode, at the top and cover it with a carbon felt, which will help to diffuse oxygen better. After sealing this cell, we'll start the oxygen flow from the top. Oxygen gas will reduce at the cathode, which will act as another electrode, and you can see this oxygen bubbles coming out of the cell. Now let's see what's the OCV of the cell. Now it is 1.42 volts. This is the battery tester analyzer that we use in our lab to test the performance of a battery. The cells are connected to it and we see their charge discharge performance here. We fix a particular current or a voltage here and we see their current, that is the discharge profile, or the voltage profiles. As you can see here, the cell has started discharging with a constant current. We can calculate the specific capacity as well as the energy density from these values. We can also check the long-term cyclability of our metal layer cells. As you can see here, there are many number of cycles that this cell has run for.
As we know that petrol, diesel and nuclear energy all are non-renewable energy. So scientists always looking for the best way to harvest energy from the renewable energy source. In renewable, best energy source is the solar energy source. Okay. For storing the solar energy source, we need two systems. One is solar cell, which convert solar photo energy into electricity. For storing this energy, we need other system which is called battery, which converts electricity in the chemical energy, and that we can use further. Having two systems increase the cost for storing the solar energy by increase the weight, cost and the complexity. So we need to improve these things. Here we are make the photo rechargeable battery, which actually stores solar energy directly into the chemical energy. So here there is no need of solar cell. This is low scale photo rechargeable battery. Here we are charging by the light, and this is the large scale photo rechargeable battery. This is in discharging mode. So this is the discharge curve. Okay, this we discharge from the constant current, and then after discharging we put light here, and it shows that after putting the light there is enhancement in the potential, which shows that this photo rechargeable battery actually converts solar energy into chemical energy.
This era belongs to electronic devices like mobile phones, cameras, laptops and all. In this field, people are mostly using silicon-based 3D materials as a semiconducting material. But to get rid of that problem, because that has already reached a limitation, to get rid of that problem, people are searching for different type of semiconducting materials like transition metal type circulars and different things. In this regards, we are also trying to grow this type of 2D thin layers in our labs using this furnace. This is the furnace we use to grow different TMDs. These are two thermocouples. This is the quartz tube. These are the controllers. We can control the temperatures from here. Then these are the channels which we can send carrier gases. This is called chemical vapor deposition process. In this furnace, what we used to do for different TMDs, we used to put different material in different temperature. Then we'll pass different gases and we'll set different temperatures. Then we'll get the film, which will be 2D in nature. To see that, we'll characterize that in using Raman techniques and microscopes. So we'll go there.
So whatever material that we are growing there, this is the film that we have actually grown. But as they are the 2D materials, you cannot see them by eyes. So what you actually need is an optical microscope. So we will put this thing in optical microscope and see what they look like. As you can see these morphologies, these are the monolayer triangles that we can grow with the help of chemical vapor deposition. So optically we cannot figure out whether it's a monolayer or not. To detect it, we usually use Raman spectroscopy. Raman spectroscopy actually tells you the energy of the vibration with which the atoms are moving. So if it is a monolayer, you can see there are two peaks. These two peaks that belongs to MOS2. These are actually the in-plane vibration and out-of-plane vibration of these atoms in this system. So what happens is, if it is it's a monolayer or a bulk. If it is a bulk, the motion in this z direction will get affected because there are multi-layers on this system. Because of this, these peaks get shifted because they are not getting enough space to vibrate. Because of that, we can actually figure out whether it's a monolayer or bilayer or trilayer depending upon the position of the peaks. After confirming the monolayers, we can actually make these kind of devices over these monolayers to use them as a photodetector or transistors or other applications that we can perform these kind of 2D materials. So I'm just testing one of these kind of devices here. So now what we will do, because we already made these devices, we will check the photo response of one of such MOS2 based photodetector, that how it will respond to the light. So now what I am doing is that I'm applying a voltage, and because of in response of this voltage, the device is showing a current. The device is here and it is showing a current. So what will happen is that the moment I switch on the laser, you can see the instant enhancement in the current as we just switch on the laser, and if we switch on the laser, it immediately start going down. So that's how these photodetectors work, and this photodetector is actually based on these 2D materials.
As we have seen, MOS2 can be used as a photodetector. So here we are going to use MOS2 as a photoelectrocatalyst for any kind of reactions. Here we can use it as a catalyst for fuel cell, where we will use light and the potential at the same time to generate hydrogen, which can be used as a renewable sources of energy. So here you can see this is a 3-electrode setup, where this one is the working electrode, where we have coated MOS2, and this is our platinum, which is working as a counter electrode, and this is our reference electrode. So let me show you how it happens. So my photoelectronic setup is ready. I'm going to add the electrolyte in my setup, and then I will perform the experiment. So my setup is done. So here you can see this is the MOS2 over my working electrode, and I have put the electrolyte. So now I am going to apply the potential. So now you can see that bubbles has started to generate. Now I am going to apply the laser. So, and you can see here the potential has started to increase. So here we have applied a constant potential, that is minus 1.5 volt, and for a time period of around 30 minutes. And you can see this one, it is giving some initial current, and where we have turned on the laser, then there is an increment in the current. That means that it increases the hydrogen generation, and when we have switched it off, the laser switched it off, then again it is going back to its previous state. That means that when we are using the light, then hydrogen generation is becoming more easy.
Okay, so we are done with the last lab tour, and let's take some questions. So the first one is from Cryptic Drosophila. Why is the monolayer in a triangular shape? Anyone from TNN's lab?
Okay, so I would answer that. So it actually depends on the vapor pressure of MO and sulfur, molybdenum and sulfur. In principle, if the ratio of these molybdenum and sulfur is almost equal, then it should be hexagonal. Okay, because the nucleation site is hexagonal in shape. But depending upon whether the sulfur is high in concentration or molybdenum, it takes a upper triangular or a lower triangular. That means the edges will end with the sulfur or selenium, and in that case it takes the shape of triangle. If the vapor pressure of molybdenum and sulfur in our CVD system is equal, it will take the shape of hexagon. Thank you. I guess I answered it.
Yeah. One more question from Cryptic Drosophila. What are the limitations of the 3D materials when compared to the 2D materials?
Okay, so to answer this, actually, why we are shifting to 2D materials is because day by day the number of transistors that we want to make in a single chip the number is increasing continuously. Okay, so but what is happening is when we are for this we need to make a smaller and smaller channels. For example, nowadays people are working on 7 nanometer channel width or 5 nanometer channel width. To go to that level, what happens is if the system is 3D material, the material that we are using is a 3D material, then there will be a leakage current. That means you cannot control that transistor at that small channel width. There will always be a background current which will show as a leakage, and you cannot switch it on and off precisely. To control this thing, we use 2D materials, because 2D materials are quantum confined, and so the leakage current will be very less in that case. So to modify or to enhance the performance of the transistors at that small level, you need 2D material. Okay, thanks.
Yeah, one more. Yeah, we have one more question. Why did you particularly choose lithium for the battery?
Hi. Hello. Am I audible?
Yes, please go on.
So, thank you for the question. So, lithium has the highest specific capacity. If you see the periodic table, lithium is the lightest metal that you can choose from among all other metals. So, it provides you the highest specific capacity, that is the charge storage capacity, as well as it is the lightest metal, and also it gives you the voltage, that is the output voltage of 3 volts. That is the highest among all the metal batteries that we have today. So this is the main reason that lithium ion batteries, they are highly successful as compared to other batteries. I guess I have answered.
Yes, thanks a lot.
Thank you.
Well, with that, we come to the end of today's program. And we hope that you enjoyed these virtual lab tours. And once when the times get better, we hope to take you on a tour of our labs on campus in person. And if you feel some people have missed these lab tours, there's no need to be sad. We will soon upload these videos on our YouTube channel. So stay tuned and have a fantastic